review, I am essentially going to go all the way back to 4.1, and I'm going to go as fast as I can and through as much as I can. I recognize that in this time period, I'm not going to be able to get through everything in detail, so I'm going to give you as much detail as I can in the amount of time, and then after school today, I'm actually going to start in reverse. So like I'm going to start with cellular respiration because that's what I'm going to end with today. So like, and then I'm going to go to photosynthesis. Okay, for 4.1, when we look at 4.1, we are ultimately talking vocabulary words. We are talking energy, kinetic, potential, first, second laws of thermodynamics, catabolic, anabolic, endergonic, and exergonic. So essentially that whole section, think matching. Okay, like vocab words are easy for me to put a matching section on the test. Now, when it comes to the definitions, all the definitions are coming out of this packet. So like your reading guide is the key to where these words definitions are. So if we looked at energy, we are talking about the ability to do work. If we are talking about kinetic, we are talking about the energy of motion. Potential is stored energy. Okay, the first law of thermodynamics would be energy cannot be created nor destroyed, only transformed from one form to another. The second law of thermodynamics is that when energy is transformed, energy is lost as heat. So heat energy. Those are probably the, 5 to the T, no questions asked about those vocab words. Okay, when we get into catabolic, anabolic, endergonic, and exergonic, those are the ones that I think they kind of merged a little together. So I want to give you some tips at how to tell them apart. First, when it comes to a catabolic reaction or catabolism, we are talking about breaking down. So we have a polymer and it breaks down into monomers. The thing that I've said to everybody today is think catastrophe. Catabolic is a catastrophe because what happens in catastrophes? Things break. Okay? If it's a natural disaster, if it's a catastrophe, something has broken. Well, that's what this is talking about, breaking of bonds. The hard part is when you break that bond, energy is being released. And that's not like a hard part. Like that is the definition. We're breaking the bonds and energy is released. But if we think of the definition of exergonic, exergonic is also energy loss. So how do you tell the difference between exergonic and catastrophic or catabolic? Look at what your test wants. It does not want the definition of exergonic. It wants an example of exergonic. So if we were looking at examples of exergonic, exergonic is a negative delta G. That's one example. It is also cellular respiration. Those are examples of exergonic. Okay, now anabolic and endergonic kind of go hand in hand as well. So when we're talking about anergonic, we are talking, oh my God, I just made 2 words into one. Anabolic, we are talking about building. Kind of like I just created my own new word. So we are taking monomers, we're building them into polymers. To do that, we have to add energy. Okay, so we're building and we're adding energy. Now, again, this wants an example of endergonic. So an example of endergonic is positive delta G. It's positive energy gain. Another example of this is photosynthesis. So that would be my biggest advice to you when you look at the test questions in terms of like a matching section. You're going to want to look for where are the examples. That goes with ender and exergonic. Where are the straight up definitions? That's going to go with catabolic and anabolic. Okay, the next thing that the test goes into, and essentially you guys, I am literally like following page for page in our notes packet. The next thing that the test gets into is the enzymes. The ultimate goal of an enzyme is to reduce activation energy. Activation energy is the energy needed to start an equation, okay? We want to reduce that. If we can reduce activation energy, we speed up the reaction. So In a normal situation, so I'm going to start with normal. In a normal situation, we have our enzyme, okay, we have our substrate, and that enzyme has an active site. And all that active site is saying is that's where that substrate wants to bind, okay? That is the indicator that this is where we should bind. Just like one door has one key in most situations, okay? One substrate has one enzyme. So they call it the lock and key model. Because they're going to fit together like a lock goes in a key. We all know that if I put the wrong key in the wrong lock, it's not going to fit. Okay, so when the substrate binds with the enzyme, sometimes there's an induced fit. Okay, an induced fit is just saying that sometimes the enzyme needs to just tiny slight change of shape. Okay, we are not talking about a big change of shape, we are just saying a tiny change of shape so it creates the perfect seal. This leads us to the last step. And in that last step, the products are being released. And our enzyme is ready to reuse. If we think about the labs that we have done that have used enzymes, we could reuse the same hydrogen peroxide as long as we didn't leave all the floating discs in it. So like as long as there's no cross-contamination, we can reuse our enzymes. Now, this is what I said was under a normal situation. On your Unit 3 review, there's a question that talks about this. It's question 4. It says, but what if? What if something comes in and it takes away the active site? So we have our enzyme. We have our active site. But we now have a competitive inhibitor. There is no way that substrate is getting to that enzyme. It's blocked. Okay? So competitive means it comes right in one-on-one, bodies up. It's not going to let you get to the active site because it is in the active site. It literally took its place. The other option is non-competitive. In non-competitive, it is the same goal. We have an enzyme, it has an active site. You have a substrate that is looking to bind. But in non-competitive, it binds to an allosteric site. All an allosteric site is saying is it's a site other than the active site. It binds somewhere other than the active site. You can see it's like literally attached to the side. You have two options that can occur. Option 1, the enzyme changes shape. The thing with changing shape, the substrate can't fit now. That lock and key doesn't go hand in hand. The second option is the enzyme does not change shape. However, that active site like gets blocked. I don't know how to better draw this because you all know my drawing skills. So in this situation, you can see that that non-competitive inhibitor, it blocks the active site without touching it. Okay, so like I'm blocking you from reaching the active site. I'm just not in the active site to do it. The last thing in 4.1, okay, so the last thing that's in 4.1, it talks about, like, we've just checked off how enzymes work. It talks about the difference between competitive and non-competitive inhibitors. The last thing says, discuss ways that metabolism is regulated, including multi-enzyme complexes and feedback. So this is, here's one way. Oh, shoot, sorry, competitive. Okay, non-competitive. There's one way we can regulate this. The last way is that feedback inhibition. And in feedback inhibition, this is where our end product goes to the front and stops it. So if I was making, let's just say, this is a terrible color choice. Let's just say this is vitamin A and this is vitamin B, okay? And under normal circumstances, everything fits. I release vitamin A and B in, and like, ta-da, they do their thing. But now all of a sudden I have too much vitamin B. Vitamin B is actually going to go back to the beginning and it's going to block it. So it creates its own block. That is your feedback inhibition. It's inhibiting vitamin B from making any more because you have enough. When you need more, that block is removed and the loop continues. Okay, questions over 4.1? I'm going to erase this, and I'm going to start 5.1. What do you got?
Why on like your unit review, there's like a lot of repeating stuff.
We'll talk about that in a second, because you're not wrong, but that's a benefit to you that it's repeated. Like, that's a good thing. Okay. When we get into 5.1, okay, and this is kind of what was just asked. When we look at 5.1, 5.2, and 5.3, 5.2 is the exact same as 5.3 except for one objective. That is a benefit to all of you. It means that I've already asked the questions once. I'm not going to keep asking the same question again. No one's got time for that. I don't have time for that. We're not doing it. So if we look at this, it says, what is the difference between light dependent and light independent? Yep, that's repeated. What are the steps of light dependent and light independent? Where are they located? What are the inputs and the outputs, i.e. products and reactants? Once I ask that question once, I don't need to ask it again. So like, as long as we can get through photosynthesis in detail, you'll have all of 5.2 and 5.3 done. That also builds into 5.1. 5.1 talks about define photosynthesis in relation to redox reactions. I'm about to talk about it in 5.2 and 5.3. I don't need to repeat it in 5.1. So like they all blend together, which is less questions for you. Now, the one thing that 5.1 does not have in 5.2 or 5.3 is explain the relevance of photosynthesis to other living things. If you flip to the beginning of 5.1, that's the answer. What is the relation? And I think most of you know this. Plants produce food, animals eat the plants, the cycle continues. So like ultimately, of course, it's not going to be that easy on the test. It is going to reference autotrophs. Autotrophs make their own food. Heterotrophs eat the autotrophs. And decomposers put everybody back in the ground when they die. Decomposers are not in your packet, but you absolutely need the decomposers because if we think about it, the autotrophs, the producers, the plants, they also need soil nutrients. If they are living in a wasteland of soil, the plants can't live. Where do the nutrients come from? Decomposers putting our bodies back into the ground. So like that whole cycle from start to finish, you could say it starts with the decomposers, you could say it starts with the autotrophs, I don't really care. Either way, it's one of those two. They produce their own food. Heterotrophs eat them. We live out our lives. We die. Decomposers put us back into the ground. Autotrophs grow. We eat them. Heterotrophs live out their life. They die. Decomposers put them back into the ground, and the cycle continues. That is ultimately, when I look at 5.1, there's your answer. What is the relevance of photosynthesis to other things? Without photosynthesis, there is no life on Earth. It's that simple, okay? We cannot produce our own food. We cannot go out and absorb sun. We absolutely have to have plants to do it. The other question that is kind of like an anomaly and doesn't fit into all of them is the wavelength. So if we are talking about the wavelength of light, the shorter the wavelength, so like the closer together the wavelength is, the more energy that has. The more spread apart, or the longer the wavelength, the less energy that has. The one thing that went right in our photosynthesis lab is the answer to this next question. What color is the worst for photosynthesis?
Green.
Green, the only color that actually did what it needed to do in our lab. Thank you, green, for not working. Okay, so if we look at photosynthesis, green is actually the worst color for photosynthesis. What should have been the best color? There's two of them, blue and red. I think blue and purple are like the same. Like I do. They're so close together in that light spectrum. So blue and red should essentially have absorbed the most light, but like we know in our little experiment, every color that wasn't green worked. Oh well, it happens. Okay, now is where we get into the bulk of the first part of the test. I guess maybe first part of the test is energy and metabolism, part 2 photosynthesis. So now here's the bulk of photosynthesis. I strongly recommend you learn these equations because the second I hand you your test, you can write the equation down, and you should. If you think of photosynthesis and you can write this out, it will help guide you with the rest of the questions of the day. Wow. That is the overview of photosynthesis. That's the big picture. That's what we need. And if you do remember that, I promise it's going to help break down in all the other things. And I'll show you kind of a little bit how. When we look at photosynthesis, we are going to start with the light reaction. And if we start in the light reaction, we are going to start in the thylakoid. And that is because it contains the chlorophyll. Bless you. We are going to talk about the reactants. The book, like the review guide, calls it the inputs. And then we're going to have the products, the outputs. Ultimately, this is where you now have to be able to break down light dependent versus independent and outline the steps that I'm about to go through, okay? When it comes to outlining the steps, whether you understand them better in picture form or word form does not matter to me. The products and the reactants are separate questions. So you have to outline the steps, and then you also need to know the inputs and the outputs. Think charts matching multiple choice type questions with more than one answer. I could put this in 2 different ways. I can give you a matching section that says, here's all of the molecules. Tell me if they're products or reactants. I can also say, here's a multiple choice question. which of the following are reactants of light dependent, or insert different cycle. And then you have to circle all of the reactants or all of the products, okay? So there are ways for me to make these, I call them Scantronable. Outline the steps. We'll see, they're a little more complicated. So if we look at outlining the steps, the first thing that we have to recognize is we are in a lipid bilayer. We are in the thylakoid membrane. Enter lipids. The very last objective of the whole unit says, where do you find lipids, proteins, and carbs? Lipids. I just don't have time to color more. So we've got photosystem 2. Remember, 1 comes after 2 because that's how it was discovered. I will do my best to give it to you in both words and picture forms. So, if I was looking at step number 1, in step 1, light enters into photosystem 2 and H2O splits. I recognize in the packet this is a little more conjoined in terms of the steps. There are more steps in the packet than I'm going to give you on the board. I like combined, short and sweet. When this happens, kind of a side product of step 1, and I don't really know how you put it in there, but a side product is oxygen is released. So like if you want to call that step 2, if you just want to call it, it's happening, oxygen is released into the environment. Step 2, which again is also like a side product of step 1, because this is kind of all happening together, but I'm calling it step 2. Step 2, the electrons enter the membrane. Because when water splits, those electrons enter into the membrane. Okay, so like all of that happens there. Step 3. Light enters photosystem 1. And because I like combining, and electrons exit the membrane. Again, we can make this a lot more steps, but we don't need to. Step 4. NADP collects electrons and becomes NADPH. There's only one thing we have left in this sequence. We have all those hydrogen ions that are kind of free floating and roaming. Remember, those are the things that go through the ATP synthase. And when the hydrogen goes through, ADP combines with a phosphate to become ATP. And we have officially made ATP. So step 5, if I had to sum this up in 5 steps. Step 5, hydrogen ions travel through ATP synthase. And ATP is made. that essentially is the light reaction. So then we can take this and look at all the things we needed. We needed sun, okay? So a reactant and input was the sun. We needed water. That was a starting product. We had water. We needed NADP. And we needed ADP. So all of our inputs come here. Well, what did we produce? Well, we produced oxygen. We produced NADPH. We produced ATP. And these two things right here are about to go to the stroma. Remember, the light dependent depends on the sun, and the independent depends on the dependent. So we need to make the energy in the top half for the bottom half. So let's travel to the dark side. So in the Kelvin cycle, we automatically can start with the two things that came from the top, NADPH and ATP. And remember, this is the only place we're going to have this P, okay? NADP and NADPH, they stay in photosynthesis. They do not come with us outside of photosynthesis, all right? Well, now here's where I said your equation comes in. You have used the sun. You have used water. You have not used carbon dioxide, which means you better use carbon dioxide. You have made oxygen. You better make sugar. We also, before we even draw this whole thing out, if you have NADPH going in, well, then you know you're going to turn it into NAD positive. You have ATP going in, you're going to have ADP coming out. So like those are automatic little things that you can just remember, because when you have one energy on one side, you're going to have its partner on the other side. RUBP is already in the Kelvin cycle, so I don't consider it a reactant because it's just there, like it's in the Kelvin. It is a part of the Kelvin cycle. Okay, I always consider RUBP, this is like our roller coaster cart. And in step one, it's carbon fixation. And yes, you need to know it is carbon fixation, and yes, you need to know what carbon fixation is. Okay, so step one, carbon fixation, essentially, here we go, carbon dioxide enters into the Kelvin cycle. So that is step one.
Wait, what is carbon fixation?
Carbon dioxide enters into the Kelvin cycle. Okay, we now take ATP. Put it in. Becomes ADP. We take NADPH. And we put it in. And it becomes NAD. P positive. All of this goes in. Step two. Reduction. At step two, reduction, this is where we have 2,3 carbon molecules. But before we get to step three, what is leaving our roller coaster ride? What's the thing we have to make? What is leaving after step two? Glucose. We've just made glucose, it's gotta go. However, in order for the Calvin cycle to keep cycling, step 3 is regeneration of RUBP. And to do that, we take our good old-fashioned friend ATP, we put it in, it becomes ADP. But RUBP is regenerated. And this is the Calvin cycle. That is 5.2 and 5.3. Not only is that outline the steps, but that is also the inputs and the outputs. If you want it. Does everybody have that so I can erase it? Whoever needs it has it, whatever you need. Okay, got you. Okay. Now, we are taking the sugar and we are going to head into glycolysis. Any questions over 5.1, 5.2, 5.3? Okay, I'm gonna count that as a no. I know it's overwhelming and I'm sure that's what most of you are thinking is just like, how do I remember all of this? Other than like repetition and staying up until midnight, which do not do that because I need you to get a good night's sleep still. I know that is some people's plan is to stay up until like all hours of the morning and then just hope for the best, but I'd rather you be energized than exhausted. Okay. What I'm about to kind of go through, just so you can follow me in terms of the notes. I'm going to start with glycolysis. Then due to where we're sitting on time, I'm going to go from glycolysis and I'm going to say there is with oxygen. So I'm going to go into the prep step. I am then going to go into the Krebs cycle. And then, I don't know why I drew such a big arrow, but I'm going to go into the electron transport chain. When we are looking at those, glycolysis, it is in the cytoplasm, because yes, all of these do say, where is the following located?
Unit number.
Glycolysis is 4.2.
I thought this stuff was in the mitochondria.
Not yet. We'll get there. And then prep step, Krebs cycle, electron transport chain is 4.3. So from the cytoplasm, now go into the matrix, which is in the mitochondria. We have glycolysis happen in our cytoplasm. From the plant to us, we eat.
Oh, okay.
Yeah. Okay, so there's the matrix. The Krebs cycle happens in the matrix.
Should we talk to Brooklyn quick? No? Okay.
And then the electron transport chain goes into the inner membrane. Okay, so yes, these parts right here, we are in the mitochondria. And again, this is all if oxygen is present, because I just think this is the part that we need to spend a little more time versus fermentation. I will still get to fermentation, but I think this is where most people's questions are coming. Okay, for glycolysis, glycolysis starts because we ate food. okay? Because we consume some type of carbohydrate. Here's where carbs come in. Carbohydrate, that's where glycolysis starts. We have a 6-carbon glucose sugar. And that 6-carbon glucose sugar needs to get broken down into 2 3-carbon molecules. And then eventually it is going to become 2-pyruvate. The unique thing about glycolysis is that glycolysis requires both ATP and ADP. So in this first part requires ATP. So ATP comes in and it becomes ADP, but that's what we use to break this down. This is where it's catabolic. You are breaking the bond. Okay? So if we were looking at the reactants for this, it needs glucose. It also needs ATP. Down here, this is again why it's unique, we're about to use ADP. So these 2 3-carbon molecules use ADP, and they do produce ATP, and they take NAD, and they produce NADH. So reactants, I need to add NAD positive. It then gives me the following products. ATP, ADP, NADH, and the key 2-pyruvate. Now, if there is no oxygen present, that pyruvate goes off into fermentation. That's 4.4. It's the next part of your packet, but I'm skipping that. And I'm going to the last part of your packet. So I am taking this pyruvate and I am sending it into the mitochondria. So we are headed to the prep step. And the prep step starts with 2-pyruvate. You can't. You'll get a WF, and that will go on your official transcripts.
What's a WF?
With fail. Your choice.
I'm gonna end that one I was thinking about.
Oh, stop. You guys are so full of garbage. Now, here's the thing. 2-pyruvate becomes 2 2-carbons. So instead of 2 3-carbon molecules, which we're very used to, we have 2 2-carbon molecules. This happens because we used NAD positive, and it became NADH. And in the process, we released 2-carbon dioxide. But we're not done. Those 2-carbon molecules also have 2 coenzyme A join in. And we have now produced 2 acetyl coenzyme A. So the reactants, we need NAD, we need 2-pyruvate, we need 2 coenzyme A. we produce NADH, CO2, and 2-acetylcoenzyme A. This is now going to go up here into the Krebs cycle. The Krebs cycle is on the very last page. It's also the citric acid cycle. So like in our notes, that's the very, very last thing. Okay. Step 1. Actually, honestly, team, I don't think the picture is helpful in this one. I think the picture is more confusing than the words. Step 1, 2-acetylcoenzyme A enter the Krebs cycle. Coenzyme A is removed. That is like a simultaneous thing. Remember how I kind of said there's no good way to put like step 1, step 2? It's not. As coenzyme A, like acetylcoenzyme A comes in, coenzyme A leaves. And you are left with 2 carbons. Step 2, citric acid is formed. Step 3, CO2 is released. And NADH becomes NAD positive. I wrote that wrong. Step 4. In step 4, we add energies. So, I mean, ultimately, I'm just going to call it that. Step 4, we've got NAD coming in, and it is becoming NADH. We have ADP coming in, and it is becoming AT. All of this is happening because we're rearranging all of these carbon molecules. So all of these carbons are just rearranging this whole entire time. The last step, okay? The last rearranging of the carbon molecules, okay, so this whole thing can start all over again, is that we have FAD. This is new. This is the new one. Enters, and it becomes FADT2. And NAD enters again and becomes NADH. The ultimate goal, the whole goal of the Krebs cycle really essentially happens in step 3. Carbon dioxide is being released. Like, that's our goal. Our goal in that whole entire thing is to release carbon dioxide. Okay. If we were outlining products and reactants. I'm gonna squeeze it right here. We need acetyl coenzyme A. We need NAD. We need ADP. And we need FAD. What did we produce? We produced carbon dioxide. We produced NADH. We produced ATP. And we produced FADH2. The last step. If we're keeping track, we have basically used our sugar molecule. We've produced carbon dioxide. We have oxygen left and water left. So we, in the electron transport chain, when we go to the reactants, we need oxygen. We also need ADP because we are about to make an awful lot of ATP. We also take from over here and we use NADH and FADH. Okay, so what is happening? If we look, this is another example of a lipid bilayer, okay, we're in our inner membrane. If we look in our inner membrane, essentially NADH is coming in and it is saying, take your electrons back, I'm out. And it is releasing all of the electrons. When that happens, hydrogen combines with oxygen to form water. FADH, which is new to us, is also releasing electrons. Then, the last thing, because the only way for us to make ATP, the only way for us to get to ATP is for what to travel through the synthase? What is the only thing that gets to travel through the synthase? Hydrogen ions. Hydrogen ions come in, go through it. We have now made ATP. So the bulk of our products, which I am throwing over here in this color, is ATP. That's the biggest thing right now, 32 to 36 ATP. That is huge, okay? Like, that is the reason for our powerhouse. Remember that. We also made water. We also made the byproducts, FAD, NAD. That is cellular respiration from glycolysis all the way through the electron transport chain. I apologize, some of the steps look like garbage, but that is the entire thing that has officially overwhelmed some of you in this classroom for tomorrow. And I'm not done.
What?
We still have fermentation.
Bro.
So,
We did it.
oh
I
yes,
have a question.
yeah, go.
So like on this paper,
Uh-huh.
It says like 2 ATP, like 4 ADP. Do we need to know?
You do not have to know the numbers.
Okay.
You just have to know where all the energies are coming and going. I will have the numbers for you. Last year's students had to know the numbers. So if you asked a last year kiddo, they would have had to have memorized every single number that came and went. That was unrealistic in my mind after seeing what they did. So now I just need you to memorize the comings and the goings. And I'll give you the numbers.
So you said when you were doing the electron transport change, you said we still need the water and all that stuff. Where did you get those from? How do you know you still needed the water?
Because of the thing I didn't write on the board. If we went up here and we wrote the equation out and we said if we put the equation for cellular respiration down, I was checking it off in my head. Which I recognize my head and showing you are two completely different things. So, thanks. Now it's in there. And that is that other piece of the equation that I said will be important. Because if you can write those things down on the test, then maybe you stand a little bit of a chance of remembering what goes where. I recognize how hard this test is. Okay, last thing. And yes, we can pull this off. Let's say there is no oxygen and none of this happens. There is no oxygen. We still need to make energy. So if there is no oxygen, we head off to fermentation. In fermentation, we've got two different types. We have lactic acid and then we have alcoholic. Lactic acid occurs in something that has a muscle. Okay, so if there are muscles, like animals galore, lactic acid fermentation can happen. Okay, when we look at lactic acid, we have our pyruvate. And then it legitimately produces lactic acid. When we look at this, it does that because we used NADH. Okay, so that's our pyruvate. It became lactic acid. In alcoholic, we have 2-pyruvate. Same concept, we add NADH. It becomes NAD. And instead we produce carbon dioxide and ethanol. Who does alcoholic fermentation? Yeast. It was our lab. Your whole lab studied alcoholic fermentation and the rate at which it changes. So, what two factors manipulate fermentation?
Oxygen.
Carbon dioxide.
What two things did we manipulate in our lab?
Temperature.
Temperature and pH. Scratch all those other things from the recording, everybody. Temperature and pH.